Hello again, this is Elim Romero, and well, today we are gonna follow up the previous video. The previous was about finding gene markers for the current sample we have. We are working currently with the cancer, colorectal cancer, adenoma carcinoma, and polyprogression dataset with just 2 samples, and I figured out something, and I'm gonna show you guys today. So far, we are gonna do this. We are gonna go and locate our sample previously utilized. We're gonna open that, load it, and just let it be. It's gonna take some few minutes, seconds, and we get this. Then remember we had these many clusters similar before. I think it's a little different. Let me update. Yeah, is the current clusters we had before, and the cell types were like this. And we verified that fibroblasts and macrophages are correct, but enterocytes and neurons we have to double check. So what I want to do today is find the markers again and sub-assign those cell types. That we have to do again into the clusters as we did before. Right, we did the clusters here, the clusters DE markers, final markers, and we got this spreadsheet here. And we had this for 6 clusters, and we have all the markers. And we verified that the markers corresponding to, let me put that back, here, I want to bring clusters, cluster ID, and bring back this. And we verified in website. Let me bring a table, sorry, a web page here. And we worked with Enrichr previously, which is good. And that's what we are going to still do to some extent. So we go to Enrichr again, and we want to just verify 4, 1, 3, and 2 in this case. And if we go and grab the markers for 1, let's take those. It's gonna be the top 20. Paste, submit. Cell types, my bad. I remember this was pointing out to something like progenitor cells, something like that. Not CellMarker, stromal cell, esophagus, cell esophagus, intestinal stem cell, intestinal stem cell colon. And we're gonna grab those markers from here. We're gonna download that again. We're gonna build the markers in this case. We gotta look for intestinal stem cell. And the 2 markers we found are these 2. We start building our marker set. Here, I'm gonna put it here. Oh, those are here already. Great. That's it. Stem cell. We have goblet cell. And we have to bring fibroblast here. And macrophages. This one. Especially we have to see which type of signature we have, so it's the strongest. So if we come back here and go to the cluster 6th and grab those again, fibroblast markers are just fine. Oh, C1K. So we do this. We paste, submit, go to cell types, and macrophage stomach human. Great. I want to see the markers here. Macrophage EC1Q, blah, blah, blah. I'm going to bring those because it's gonna make easier my annotation. So we look for macrophages, stomach human. We bring this set and we copy paste here. Back, close that. Here, we are going to replace that here. And we are gonna do one thing after just to verify. I think progenitor is gonna be still fine. We are going to figure out that later, but fibroblast and macrophages gonna stay the same. So we keep those for annotating. We can erase these two for now. And let's keep building. We look at the cluster 1, which was stem cell. And if we come back here, we want to go now for cluster 2 here. Let's see what's in there. Here, again, cell types, goblet cells. That's gonna be the goblet cells here. Large intestine human, looks good. So far, so good. Let's get the table. That I think we have it already, but let's bring it on. Goblet cell. So we get these two, and we can, oh my god, that's not the one. And we can bring it on here. These are the two marker sets, the two marker set. Then we gotta go back, and let's look again now for cluster 3. Cluster 3 is this guy. Cluster 3, paste cell types, and this was MK67 progenitor. We can just call it progenitor cells, but let's get the markers. Progenitor cells. Here I got this, and neural progenitor here. If you see this and this marker is the same, this and this, so some of them are repeated, which is great. We can be sure that those are similar or very conventional progenitor cells. So what we're gonna do again, it's copy this into progenitor. I think it's the same. We build, we build, we build, and that's it. We are gonna use this set to verify things later. Then, so far, so good. Now cluster 4 is the only one we didn't spend much time before identifying. So we're gonna grab it, come in here, paste, cell types, and immune cell. No, it's not immune cell. Plasma cell breast human. No, it's not either. So we gotta fish around and see if there is something here. I think those still gonna be progenitor or stem cells. So still we can look around and see if intestine large. So it's but it's mouse at last. Gonadal, microglial. Let's go for the CellMarker 2024 and let's see what is in here. Immune cell. Here is a trick. We can look here, colon, glial cell, stromal cell colon. So very likely no. Then the problem is that now we have to WNT2. That's a gut marker. Let's see. No. Monocyte is not either. Fibroblast is not because it's separated. Potential is not. Stromal cell, macrophages. So this is gonna be a hard cluster, and that might be because it's already repeated to some extent. If we remind, we already identified the rest. So if this comes in hand, that means that that is complicated. And the trick we have to do here is to select that cluster for and subsample. So we are gonna hit here and select by class, cluster ID, okay, alphabetic is fine. I'm gonna grab the number 4 and proceed with the selection, and it's gonna bring it on here. So here, what I can do is do the clusters again. I'm gonna store it. So this is gonna be now the SCE1. I can close it. And now I can do SCE1 equals to latent cluster SCE resolution 1.5. We can use 1.0 or 1.5. 2 is too much because it's too few cells. So we do that. We let it be. Run it again. And gonna stop the video for a few seconds. Right, it finished. So oh my god, I screwed the thing. I overrode it into SCE1. Wait a second, I'm gonna correct it. Okay, it didn't happen as I wanted. I wanted to subsample this cluster and recluster, but what happened is I recluster everything. But I already feel comfy with the markers for this set and these other 2 ones here. So the only thing I have to focus is in this other population here. So I'm gonna do again the marker, define all markers on the cluster ID. And in the end, I'm gonna look at that set later. So I'm gonna stop the video and come back once it finished. All right, it finished. And remember, we get the table, we look at the very end. We have 15 clusters, and previously we have 15 clusters. So we export the Excel into the all markers. Here I did something already to the other set. I already named it as 6 clusters and 6 clusters, so we don't erase that and we keep it just in case. And this we call it 15 cluster or clusters. Okay, we close, and again here we need to rename and we copy that name here and paste here, and that should work for our new set. So we select all and run, right click and evaluate. Gonna give it some few seconds. Oh, it finished. Great. And then we look for that. It's gonna be this one. It's gonna be the top 20 markers. But remember, now we are only focusing in this fraction in the left. So what we're gonna do here, it's again do this this way, minimize this a little bit here and there, get this body here. All right, we get this. Remember, we are populating here in the bottom, and I'm gonna discuss something in the end about that. So we focus in cluster 8, 13, 15, and 12. So let's go for 8th. 8th, it's gonna be this. Ooh, I've seen some of those markers before. We submit, and we look for, come on. And we look for cell types and goblet cell. Potentially it is goblet cell, mouse, goblet cell, colon, human. So those are goblet cell as well, but those are different goblet cell. Those are coming from different genotype. But let's look around. What else is there? Panet cell is possible. Goblet cell again, lighting up here. Goblet cell here and there. Yep, it's goblet cell. So we can look at this table again and export table. Look at that, opening it, and we look for a goblet cell. It's gonna be the top. These are the ones, and here's a trick we can do. We can call it goblet 2 and paste the markers. That's it. We are gonna keep it like that for now because I think those are a little different genotype. So now we are gonna clean these others and we proceed to look at the other. Let's look for 12. 12. This one's not so sure. Let's look. Fibroblast. Yeah, I think those are fibroblasts. But lung human, stem cell kidney human. It's possible it's a stem cell. Let me see the markers. No, I don't remember any of those. Let's have another glance. Mentions here vascular endothelial. I don't think those are vascular. Cancer stem cells. But uterus. Be careful. We gotta be very careful. Stromal cells. That might be potentially it. Adipocyte, fibroblast, kidney. So we come back here. I don't think it's cancer stem cell because remember, that's healthy sample. We show the batch ID. A014 is unaffected polyprogression, which is still not cancer active. So I don't think we have a reason for naming that cancer stem cell. So we reject that because I don't think it is. But it's potentially fibroblast, a different fibroblast. Stem cell tumor infiltrating T cell. No way. No way those are infiltrated cancer yet. Let's see what's else here. It could be large intestine progenitor, but different lineage. So what we can try right now, as we have epithelial cell, no, I don't think it's that. Fibroblast, I think it's potentially fibroblast because I remember having fibroblast before with the PanglaoDB. If we just re-annotate here, with the PanglaoDB, I think it has to light up as fibroblast is 12. I think it's a 12. But I don't know what type of fibroblast. Right, it's there. It's coming out again. Here. So this is also fibroblast, but it's different fibroblast population. So we can call it fibroblast 2. And in the end, we resolve what it's different within it. So what we can do is repeat, come in here and look for fibroblast. Although it says ovarian, but it was fibroblast somewhere else. Fibroblast. So in theory, that other is also fibroblast, but it's different. What can we do here? I want to try something I haven't shown. If we grab these markers and annotate the customized, so we take out the cluster IDs and annotate customized markers, no, and then copy this, copy and paste that, and yes, automatically I think we're gonna have some interesting results here. Excessively strange. We have stem and unknown, so that means these stem cell markers are not great. We got to look for something better here. Although I haven't looked at the expression of those, and this is something I didn't show before. We can do this expression into the UMAP, and I'm gonna show you something different in short. So it comes out this one. Okay, it's everywhere, but mainly in the right hand side. So I don't know why it call it stem cell over the place. So something is happening here. Then I mentioned before that could be contaminated, and we gotta be sure. Oh, PBX1. So what we are gonna do is we are going to remove this one and include LGR5. That's a classical stem cell marker, and we can utilize that to annotate. The reason I'm removing it's because it's everywhere. That's not informative. So if we do this again, potentially we could get something different. Cluster ID, we show it again and re-annotate. Annotate, copy this, get it here. Yes, automatically. Now the only right thing is this fibroblast. What's that? Progenitor. That should be macrophage. That should be macrophage. That means some of these markers are not helpful. So we gotta see which ones are not. Very helpful. Paste. And look at that in the UMAP. And we get this. Progenitor ASPM looks good. This CNF looks good. This INPP5D doesn't. NUF might be fine. So it seems that the only noisy marker is this one. So we start refining the marker set. So if we try it again now, annotate, customize markers, paste it, and yes, automatically. Okay, now everything is unknown. It's not identifying my macrophages, and that's not great. So I'm gonna verify the macrophages markers here. Yes, A and B. And here's gonna be one cool thing as well, but I'm gonna do it in short. C1Q, it's a great marker, but it's not showing much. CHIT1, those are good markers, but it's not showing much of expression. So this is lighting up the two things, that I filter up too many cells or that it's poor quality. So it can be either. Now I'm gonna use my previous markers and see if any of those light up properly. Paste. Then we get this. Nope, it's not lighting up nice. That maybe I'm gonna bring it to the macrophages I'm building here. Then CD68 is not helping. MRC1, it's kinda and kinda not, but we can keep it MRC1. And what we can do is, again, do the annotation, but we get to light up the cluster ID, annotate, and then we copy paste fibroblast, and macrophages didn't come out again. So here our alternative that building our clusters markers didn't work. We are gonna do a different venue. So we are gonna do cluster ID, and we are going to annotate with the Panglao automatic. So it's gonna be an iterative refinement. So the thing is that there are some gene markers that are not expressing too much. That's the main problem. My bad. Cluster ID light up. Okay, and now annotate. Panglao, human, yes, automatically. The problem is that it's gonna tell neuron cells. But here's gonna be a nice trick. From here, we're gonna do again the hit marker, but in this time the marker gene heat map. Sorry. This heat map, do not only consider the Panglao gene markers, cell types, method 1DE. Then we just let it finish. Manually order groups, no so far. Yes. Normalize A and B. Row-wise normalization. What I usually do is range 1 to 10 for subcell types, but here I think is standard my work. Now it fell. Too many inputs. Okay, let's try it again. It fell, so needs to be debugged again. We do cell type, this, manually order, no, that, and row wise 1 to 10. Let's see if that works. Let's give up on this. So that's it. Now another venue we can do. I'll have to show the gene marker heat map later. What we can do here, it's look into the cell types. Done. Gene expression, and we're gonna paste a gene list. For example, I want to see who is more macrophage. So I do it. Yes. A and B. And then we get it. B cells has more macrophages. So it's wait, we had macrophages here? No. It's annotated as B cell this time. Right. It's detected as that cluster. So there is something wrong in this cluster. Should be another different cluster. That's not identifying it properly. So we gotta do again the clusters. So let's do it. We're gonna clean the environment, and we are gonna load the noQC, and then the SE equals to the laden, and resolution, let's put it in 1. Yeah, 1 is fine. And I'm gonna stop it for now. Okay, it didn't work with resolution 1, so I had to use 1.5 resolution, and gonna stop it again. All right, it finished, and now we have this cluster 19 just separated. So we are going to do again the Violin plot per cluster ID in this case, and here we're gonna grab only the macrophage markers. Done. Gene expression, paste our gene set A and B. Let it be. Gonna stop it for now, and we got it. Here we got to start inspecting what is potentially there. For example, 19, it's pointing out as I wanted before. That's macrophage. Great, 19 is macrophage, and that's it. Is the only one. Should be, yes, the second marker is lighting up in that. This one, CD84, we have to remove it because it's lighting up also the 17th. So that's another immune cell marker. So let's go and look for CD84. We remove it, and that's how we curate. 19 looks good. This CSF1R, it's a good macrophage marker, and we can remove MRC1. So if we do this again, I'm gonna close it and do again the Violin plots per cluster ID for all the clusters, genes, and copy paste A and B. can stop. Oh, I didn't have to. Oh, I'm gonna just wait. It's not gonna take that much. And we get it. Again, this is one, this is the second. Okay, great. Everything is lighting up properly. So now we have a way better curated macrophage. Now, we're gonna look for fibroblasts. We had two populations before. Now we have to figure out if we have it again. So Violin, cluster ID, done, gene expression, paste, normalize A and B. It's gonna take some few seconds. And we get it. And let's see what is lighting up. 15. 15 was fibroblast before. Yes, 15 and 19. Where is 19? Here. So that's not what we would like to, but still is very pronounced in the 15. This, oh, that's great. Col 3A1, it's great. And we can remove the Col 6A2. So we can remove this one. And let's see if we have some other, no, we don't. We can keep this too. That should be good enough with this and this. Very likely just the Col 3A1, but we can keep the rest. And again, let's look for stem cells. And we're gonna do the Violin plots for all the clusters again. Done, expression, paste. And here I'm gonna do something I haven't shown you. I'm gonna go for the specific stem cell markers. I don't think this help much. 14, 10th, number 5, 3, 5, 3 should be lighting up. 5, 3. I think if we, oh no, 11, yeah, very likely 11 or is progenitor cells. So the 11 has that very well marked. And here I'm gonna do a trick that I haven't done before. So I'm gonna go to Enrichr and go to libraries and look for CellMarker 2024 and download that database. Might take some few minutes. And we are gonna look for one specific intestinal stem cell. So stem cell. And we keep looking. Cancer stem cell. Here we have potentially cancer stem cells, but not right now. So I'm gonna grab these markers and I'm gonna see if any of those work. It's tricky because we have colon, intestine, and the rest it doesn't matter. But I can try this marker set, this marker set. I'm gonna bring those ones to my sorry for that, for this guy. So we're gonna keep it here and let's fish for some other stem cell, breast, colon, human. Here's more. We have even more. Copy and paste. Colon, cancer stem cell. Okay, we can consider that. Cancer. Wait a minute, the other word, cancer? Cancer stem cell. We can consider those, but we gotta look for normal stem cells. Cancer, embryonic stem cell, skin, progenitor, intestinal. This is very likely what we were looking for. Epithelial, not mouse, but human. But if you're working with mouse, this might work. So I'm gonna copy this intestinal stem cell, small intestine. No, we are working with colon, large intestine. And come back here, intestine mouse, small intestine. Let's see if we find some other more. LGR5, large intestine. This is something usually what people look for, so we can consider that. Let's keep looking for leukemia, memory stem cells, and neural stem cell, pluripotent stem cell. Basically, whatever it brings colon and a stem cell, we can utilize. But most of the time, we gotta be careful how we curate this data. In a adipocyte, no, none to this. Brain, colon, human. Here's again. So I'm gonna bring it. Oh, all these markers I can try. Very likely this is the one I was looking. It has LGR5, OLFM4, SMOC2, SOX9. Potentially that's what we are looking for. And intestine human. So this is another set, and look, it's similar. So that's what I like, that you have little different, but you have very similar markers. Axin 2, I've seen it, ASCL, PROM1, LGR5, CD44, maybe not, but we can see what we find. Embryonic intestine. Here is this other human. I think we took it already. Keep it going. Beta, activated neural, adipocyte, cancer. So we already turned around. And now we are gonna look at those markers, but we got to do it in format. So I'm gonna stop and format it. Okay, I formatted that. Unify the cancer stem cells, just not to repeat it. The colon. This is gonna come weird because many of the stem cell colon, just a stem cell, are repeated within the cancer stem cells. So that's gonna be tough task, but can be done if we do it carefully. And I'm gonna raise the stem cells here in the top, and we can try this set again and see how that goes. So we do annotate, markers, no, paste all the markers we have, and just automatically, please. Yep, it was bad. It was bad. Well, interestingly, we found cancer stem cells known in fibroblast. So we lost this section by cancer stem cells. So that means that way too many of those are also expressing in that cluster, and we got to find out which ones and remove those. So in the cluster number 19, so we're gonna go and look again at the Violin plots for every cluster, but we are gonna focus on only into the cancer stem cells. And let's go and look. Done. Paste all the markers, yes. A and B normalization. Let it be. Gonna stop it. Okay, came back, and this is what we get. And now that I remember this first marker, ITGB1, I think it's a macrophage marker, so that's why we are getting incorrect annotation in the top. But there's one thing I did before. For goblet cells, I just remembered that MOC2 is a good marker, so just to verify MOC2, include tumor samples just in case, and is a goblet cell marker. So I included to the goblet cells. So this first one, let's go and see what that pops out. Unknown fibroblast. I thought it was macrophage. And here's another trick. Gene marker. Just ask, and I think Gemini is gonna answer. Which cell type? Which cell type? Just in case. And the variety of cells. It's fibroblast, myofibroblast, endothelial cells. So it's not a good marker, in short, because that's fibroblast-like. So we remove that. And we have to curate this gene list. And here's a trick I'm gonna show you. We're gonna go to Gemini, and this is because I'm feeling lazy, and I'm gonna ask, hey, do you know which markers of these are cancer stem cells? Oh, my bad. Cancer stem cells? Oh god, come on. Copy that and bring it on. It's not copying? What's going on here? Okay. What about these for just stem cells? And then we're gonna ask for that gene set here. And what about LGR5 plus stem cells? Here in the bottom. I think it's too many, those LGR5. That I don't think it's good. So it verifies, oh, this is a good set. And I think it starts telling, oh, it's in various cancer types. CD44, 24, and the reason is surface. This is definitely LDH associated with detoxification in many solid tumors. PROM1 also is a good marker. LGR5. So here, what we're gonna do is graph this curated for just stem cells. Just stem cells. I'm feeling lazy at this point, so it didn't change. And we're gonna grab this for cancer because many of these are not correct. Let's see if that reduces. Yeah, it did. Good. And in theory, it's getting better. And now about LGR5. So just these three markers. Well, and TMAM. Just in case, we're gonna try to annotate that. I don't think it's good because we have already stem cell and that requires LGR5. And let's do again the annotation. Once again with this refined gene marker set. So we go annotate, customize, no, and then do it automatically. Fibroblast, unknown. At least I like it, it's unknown at this point. So it didn't identify it as, and this is one problem. This is getting better. This region in the bottom was progenitor. It has MKI67. I can prove it. Paste. MKI67. I'm sorry for that. Yes, normalize A and B. And yeah, in the very bottom here. Yeah. So, and progenitor cells are also expressed in some other type of cells, and those can be in cancer stem cells. So far, I feel not confident, but a little bit good about these gene markers. I'm gonna try to make it easier because I think too many markers is confounding the algorithm. So I'm gonna try to just take all these other, and I'm gonna try to find out what else is there in macrophages, if there is anything. So we're gonna go back again, cluster ID, and then annotate with customize, no, paste the markers, and yes, automatically. I'm about to give up on this, but not yet. So it's the same. So that didn't help much. So actually, I can remove this stem cell and just keep the LGR5 stem cell and see how that goes. And here is something I will do. and it's a good trick to work around this issue. Cluster ID, and annotate again. Let it be. Now it has less stem cell markers and should confound less the sample, and it classified as a stem cell progenitor. So we are not getting the stem cells. So the OGR5 is not helping. We're gonna get just the stem cells, and goblet cells not being identified at all. But I think it's fine. I think it's just fine. The issue is that this is confounding the rest of my markers. So what I'm gonna do here is make stronger the fibroblast and the macrophage markers. How? I'm gonna, with the brush, I'm gonna select just this region, which I know those are macrophages, and do here find marker genes for brush cells, lasso, and brush cells only. Top 20 is usually working. Sometimes the first 10 it doesn't tell much. And we are gonna look for the gene markers, but now that express specifically in that region. And this is what we get. Itgax. I think that's a good marker. So we have to now go back and verify in Panglao. Itgax. Tell me, what's that? Dendritic macrophage. Good marker. So we can bring it here into macrophage. Let me close this. Let's go back. That one, not good. That's a good one. MSR1. I don't know. I haven't seen this marker, so we're gonna figure out. Dendritic macrophage. Great. If I use that, it's gonna help. Let me close this other. And step 1, I don't think that is a good marker for this. Macrophage, right. It's an activation gene. So far, we have to look at what's in there. Sep2, I think it's good. Let's find out. I've seen that guy before. Dendritic, oligoglial, macrophage. So it's immune cell. Very likely not helping because we have T cell, NK cells. Belongs to many cell types. Not helping. And I don't think any of these others gonna help. Right. Maybe let's look at this one. And neither. That's dendritic cell. So we are gonna take this gene set and let's see how that turns on in the clusters and see if that helps. Again, come here. Cluster ID. And then we're gonna plot the Violin on clusters. For all of them, gene expression, paste that gene set, A and B normalization, and we let it be. And we're gonna get the markers now into every cluster, and I wanna see if all of them are lining up in the 19. And oh god, I shouldn't have done that. It. C1K19, good. That too. This is also good, and good. That's gonna help us. MSR1 is great, so that's gonna get definitely now the macrophage. Although we couldn't get the fibroblast, the goblet cells, but that's fine. That's fine for now. Just demonstration how to curate back and forth. We got macrophages now. Nice. Now there is a problem. Here, I couldn't find anything. And these unknown cells still need to be clarified. But this is looking good in the right. Unknown cells here. And what we're gonna do again, it's going to cluster ID. And now we're gonna do the brush selection. And we're gonna look for the markers, lasso regression, brush cells only, the top 20, and see what brings out there. I think those were fibroblasts as well, but we gotta figure out what other markers are in there. Okay, that's really lighting up. And let's ask Panglao. Germ fibroblast. Okay, that's fibroblast. If we use that marker now, here in the fibroblast 2, now we might get it better. Now let's go back here and there. That I haven't seen it before. This is lighting up good. Let's see if it is related to fibroblast. Nope, it's neuron. Wrap, it's also so-so. We can go and look. Enteric neuron, it's lighting up that is neuron-like. I do not recognize any of this. So we go for this, paste, nothing. It's getting weird. Robo1, I've seen that guy, but I am not sure what's related to that gene. Unknown, yep, it's just something that pops out. Papillin, TNXP might be something. Fibroblast, perfect. This is one. And let's go for TNXP. Find for it. Search. And germ cell. That's not what we want. And that's it. So we only could rescue some 2 out of the 20 or whatever we could do. But I think now we should be able to get that other fibroblast population. And then we gotta do the 18th brush lasso brush cells. Top 20. Then we get this. I'm gonna come back to that. Meanwhile, I'm gonna try to re-annotate. And here we go. We try the new marker set. Uh-oh, didn't get the fibroblast there. I thought it will bring it, but it didn't. All right, so far I'm gonna roll back to this. And I'm gonna do look for the 7th cluster. Maybe like this, right? And I'm gonna do the DE brush markers. Top 20. I'm gonna stop the video for now. Oh, it finished. Okay, this is lighting up that Mach 4. That I'm not so sure what's that about. Let me keep it like that. So we come back here, paste Mach 4. Epithelial cell. Yeah, maybe. We can just use epithelial cells. Yes, cell. Mach 4. It's not what I want, but it can do the job. this one, oh my god, that's very expressed. And let's see what that is. Fibroblast, plasma, B cell, dendritic. Nothing that I can relate. It's not something specific. So we're gonna go. It's PDF. That is goblet CellMarker. That's goblet CellMarker. Yeah, that's goblet CellMarker. So I can bring it to the goblet cell itself. Potentially those are goblet cells. DDP, let's look for that. No. What about this? I didn't copy. Nothing came out. Okay, let's keep looking for cap N9. That I've seen it before. Let's see. Goblet cell, perfect. We can bring it on. Goblet there, goblet here. Colon, which is great, exactly what we want. PCK1. Let's look for this. Epithelial cell. So I'm gonna bring this to epithelial just in case. But this also goblet cell. So I'm not gonna fight within epithelial. I'm just gonna put it in goblet and in goblet here. And those are Mach 2. Mach 2 is there. And Mach 2 is here. Let me bring it together so we can correlate faster. And I think that's it for that one. CK. Let's see what this final is. Luminal, basal, epithelial. That's it. We are not going to use that one. So far, if we use this, we can close this one. Oh, close it. And re-annotate. Annotate again with customized markers. Yes, automatically. Still got it unknown. I was expecting that to be goblet cells. So this unknown can be potentially also goblet cells. All right, then we got to go back again and clean it up. For now, what that cluster was, I just want to see. Cluster ID. What cluster was that? Gonna close and redo it. The 18, right? Was the 18? Top 20. And let's see what's in there. Okay, very few. I don't think. Okay, that might be fine. Let's find out what that gene is. Enteric glial cells. Enteric. Easy. Enteric. What that is? I forgot. Easy. We can just label there. KDR. Let's see. Easy. So we keep it. Let's look for this other that rings the bell. Come on. It's not KDR. EC as well. And so forth. Let me do one last gene and we are gonna call it a day. Let's look for CDH5. That also rings a bell. Easy, perfect. So we are gonna use this one as well, and I can do things here, but I want to try this automatic annotation to see if we can fix it to the best we can. Okay, that light up as easy. That's a good gene set marker. And merge. And that's how we curate little by little the data set. So now, for example, here, doing the brush markers and so forth. And this is exactly how we build the clusters, sorry, the how we build the, let me just light up this, how we build the cell atlas. And this is finding the markers and annotate. There's one trick more. If I want to hard code assign cells, I can go here and I'll let it know, assign cell types to groups. No, maybe not that one. Well, I'm gonna give it a last shot here. You see all the neurons were either cancer cells or stem or progenitor cells. That's confounded most of the time. Oh, we have B cells. I can try my B cell markers here. Yes, merge. And this is enterocytes. So here, to find out what gene markers are here in enterocytes, those are epithelial cells as well. And these are fibroblasts, as we mentioned before, but those are different fibroblasts. If we do look for, in this endothelial were ECs, not endothelial, so that means this small branch is endothelial, but these are ECs. Oh, the ECs are endothelial cells, my bad. Well, now if we want only to get the markers for these cell types, we do here find all markers for cell types, and we let it be, and I'm gonna come back in short. Okay, to finalize, I want to show, after using PanglaoDB as well, this is iterative. You gotta go back and forth in all the places because building the cell atlas is complicated, and we gotta get just the exact markers that our dataset needs. So what I'm gonna come and get from B cells is just the top maybe 5. Indeed, I can double check that those are B cells markers. These are B cell markers from our dataset. Let me extract them. I just have to do that. Coming back soon. All right, I couldn't extract it, but what we're gonna do here is just sort by group, by fold change, and p-value in this order, largest to smallest, smallest to largest for p-value. And we're gonna look for the markers. So these are the top 5 markers, 20 markers. Cell is a good marker. And we can go back and go to Enrichr and verify that those are good and valid B cell markers. Just go back and forth. B cells, B cell kidney, it's fine. B cells memory, B cell naive, it's fine. PanglaoDB, where is PanglaoDB database here? B cells kidney, it's fine. Spleen, B cells. Where is PanglaoDB? PanglaoDB is not here. So, long story short, we can just see which ones are the best. For example, I can grab this first B cell markers. For example, the markers I had before are not lighting up here, not at all. So very likely I have to customize the B cells here. But I can evaluate with this. So oh my god, MS481. Okay, and there is another epithelial. I want to grab the epithelial markers from here because those wasn't so sure. I'm gonna go to enterocytes. Those are gonna be my epithelial, And I'm gonna grab these tops and format. I'm gonna come back. Okay, after grabbing that and setting that into Enrichr, it points out that is epithelial in many places. Here in the augmented, and I think also here epithelial. That's not exactly our tissue, but I think it's lining up different epithelial, intestinal cell epithelium. So those genes here are good to use. And I formatted already, so we can utilize this final set and see how that performs. So we do cluster ID again and annotate automatically. Okay, it's a little better. It's a little better. So that's it. Although this epithelial should have also stem cells, but those gonna be different stem cells. Here are cancer stem cells associated and progenitor cells. But this is not that bad. This here, the unknown, might be epithelial, just epithelial cells. And so far so good. That's what we got for today, and we built this atlas. It is complicated, but it is what it is. All right, thank you for seeing the video. See ya.